Yes, perfect. So thanks for the organizers for inviting me to give this talk. I'm not sure, maybe not everyone was at ISMRM. So we'll go over it again. So again, this work is looking to combine MRI and histology metrics. So my expertise really is on the MRI side of it. For the histology, I really have to thank my co-authors, Darian Chitzaz and Timothy Kennedy, for really bringing the staining aspects of this project to life. So we're interested in the common marmoset, and maybe why are we interested in imaging the marmoset? So it's being rapidly adopted by the neuroscience community to help us better understand the human brain, really helping to bring and translate the information we know from rodent models towards humans. So it's really being picked up for that. It's an ideal model for human brain imaging as well, because it has similar cortical myeloarchitecture patterns to humans. So for me, I'm really interested in intracortical myelin, so that's a big bonus. It also has a thick and smooth cortex, as you can see in these images here. So not that much gyrification, which can be great for a lot of things, including limiting partial voluming effects, which you see with very curved brain sections. And also, if you're interested in the white matter, there's also a lot more white matter than in the rodent brain, so that's also a good plus. Because of all of these things and some more, that's led to a generation of many marmoset atlases that have been pushed out. So I mentioned there's a lot of marmoset atlases. We don't have to go through this whole list, but really what we're highlighting here is the recent growth in this field of marmoset neuroimaging. And there's the original or an early paper by Paxinos in 2012, but since then, within the last 10 years, there's been many papers publishing open atlases on different parts of the marmoset brain. And what I really think this group should look at is there's high impact journals here. So Neuroimage, Nature Communications, Cerebral Cortex. So the neuroscience field is really appreciating and rewarding the efforts that goes into these kind of projects, especially when this data is made open for other researchers. So I think this is just something that we should be keeping in mind as animal researchers in increasing the impact of our work. So where does this specific atlas fit in with that whole list that I shared that wasn't necessarily exhaustive? But we have some uniqueness. We have a combination of microstructural MRI contrasts acquired that is unique. So many of the previous ones, very limited, some very good diffusion data exists, as well as some light MT work and some T1. And T2 weighted tends to be used for an anatomical. Additionally, instead of using an animal scanner, this project was really focused on translatability. So we used the human system, so using the same hardware that we would use for human imaging. And additionally, we have several stains that weren't previously acquired. So again, in many of these atlases that exist, there's a lot of Nissl stain. There's some good silver myelin stains as well. But as you'll see, we have some that aren't previously available. So the initial goal of this data set is to validate the sensitivity and specificity of some new, like IHMT, and commonly used, maybe like R1 microstructural markers in the brain. So what are we interested in with the brain microstructure? So for me and a lot of the neuroimaging field, it's myelin. And this recent meta-analysis by Mancini et al. Just goes to really highlight, if you look into the field of neuroimaging, what is being used to look at myelin content in the brain? So we have a wide range of diffusion imaging, which are these purple markers here, to magnetization transfer. So green, we have all of these metrics, T1, T2 relaxometry, as well as some other markers. So this really leads you to wonder, what should you acquire if you're interested in myelin? What's going to give you the most sensitive marker? Maybe you have something in the brain where a lot of things are changing. You'll need something more specific. So really, what should you be acquiring for your studies? And this might be study dependent as well. So for this study, we did in vivo MRI in a common marmoset over 4 sessions. So each session lasted roughly 2 hours and were spaced 2 weeks apart to ensure good health of the marmoset. So some of the contrasts we acquired here for anatomical imaging, we have R1 quantitative maps, T1 over T2 weighted imaging to compare against maybe human connectome project style acquisitions, MT-SAT maps, so magnetization transfer saturation, and then we have T2 star-based myelin water fraction, T2 star, and IHMT-SAT. So the first 3 were acquired at 300 micrometers isotropic resolution, and the last three were 450 micrometers. And these images on the side show the chair that the marmoset was imaged in. So they're placed in sort of a supine position in this chair. So this isn't the coil specifically, but we have a specific 16-channel marmoset coil where the head would fit in right there. And for the images acquired, they were all, this chair allows them to be rotated a bit in the scanner, but we had them all fixed at around 45 degrees. And we don't have to go through this whole chart, but in case people weren't at the ISMRM or don't have access to my abstract, this is sort of the information that was shared there, and it will be shared in the YouTube recording. But we use a 3T Siemens PrismaFit scanner and a 16-channel marmoset RF coil for receive. So for the T1 and T2 weighted images, we had a turbo flash and space sequence, and we did variable flip angle T1 mapping, and then used a GRE scan with typical MT-SAT parameters for the IHMT. So like, if you're not familiar with this, this is a marker that aims to be more specific to myelination patterns, where we acquire several magnetization transfer weighted images a little bit further off resonance than typical MT, which is 2 kilohertz. Here we're 7 kilohertz off resonance, as well as MP2 range and multi-echo GRE for T2 star and myelin water fraction based imaging. For the histology aspect, we acquired block face images to help with the registration. So we sectioned the whole brain on one hemisphere, and every time we took a section, we took a photograph from on top to help with this registration. So here's an example photograph of the brain. And we acquired, for this work, presented today, 3 stains. So we have host for the cell nuclei stain, ferritin, which is an iron storage protein, and we use fluoromyelin, which is a stain for myelin and membranes. And this was acquired with a 10 times objective lens for a pixel resolution of 0.67 micrometers. And just an example of what the fluoromyelin stain looks like. In part of one section, we see these tiling images as the microscope scans across the section. So a little bit more processing had to be done for histology. So with the block face images, like I said, we used that to help with the registration. So in order to do that, we needed to reconstruct that into a 3D volume. So we did that with this POSSUM framework, which uses ANTS registration. And ultimately, we end up with an initial 3D volume, nifty volume that we can use for registration. And then for the histology, we had to correct the vignetting in the tiles, stitch them together, and with the host stain, we did a rolling ball filter to remove some background fluorescence. So to combine all the data that was presented here, again, we used the 3D block face as the target for the registration. The MRI was registered with 3D, it's a fine and rigid registration, and the histology was a 2D registration because the slices were matched with the photograph, so we knew where they were. And then from there, we did manual ROI delineation and ITK snap, and the metrics were correlated with the MRI to the average fluorescence intensity of each of these markers in the ROI. So looking at some of the resulting figures, so here we see alignment of the 3D MRI. So we have our 6 MRI contrasts with a single slice look used in this analysis. And we have a tiled image here that shows this alignment. So the block face reconstruction really helped with this alignment. Maybe easiest to see if you try to follow the white matter track around. So in this tiled image, we have the block face images in pink, the MT-SAT is grayscale, the ferritin stain is red, and the fluoromyelin is green. We can also see maybe where this myelin water fraction maps are not fitting very well. We have some susceptibility artifacts that are most prominent in the posterior regions and definitely inferior regions of the marmoset brain. And once you see them here, then you can also point them out a little bit better by darkening in this T2 star map. So for this analysis, we picked 5 sections across the brain. We do have sections across the whole brain, but for this limited analysis, we only picked 5. So we see good staining homogeneity across the sections and between sections as well. So all of the stains were done by a submersive method. So all the stains were in for the same time with the same concentrations. So we could see good staining here, and the ROIs delineated. We have chosen for white matter, subcortical gray matter, and cortical gray matter. The colors might seem similar here, but each section was had its own ROIs. So if we look to the results, we see high correlations with all 6 metrics with the fluoromyelin stain, which was we were using as a myelin marker. So the correlation coefficients here for all of them were above 0.9, so very high correlation. And this was expected based on the recent meta-analysis and maybe corroborates many other studies suggesting that their MRI contrast is indicative of myelin content. So if we try to get some idea of specificity, we would want a metric where the MRI metric hits 0, where the y-intercept is also 0. So if we look to the lowest y-intercepts, we see IHMT sat and myelin water fraction present very low y-intercepts. Both of these metrics are being touted as being myelin specific. Surprisingly, the T1 over T2 weighted had the absolute lowest y-intercept. So this might suggest specificity, but we'll see in the other slides that might not be the case. Across all of the section or the contrast acquired, they all had weak sensitivity to nuclei, with R2 star presenting the greatest correlation with 0.22 as a correlation coefficient. So interesting to note here is they're all in the same direction as the myelin correlation. So this could be a potential artifact of spatial colocalization. It'd also be interesting to follow this up with comparison against a neuronal nuclei stain. And there's an atlas that already exists in the marmoset for this, to see if there's specific cell types contributing to this. For example, all of these positive correlations rely on the white matter points, which are slightly above the gray matter ROIs. And what we know about the brain is there's very little to none neuronal cell bodies within the white matter. So these data points here are primarily indicative of glial cell density. If we move on to the final stain shown here, we have differing sensitivities to ferritin. So highlighting the first one, T1 over T2 had the strongest correlation with this iron storage protein. So if we go back to the fluoromyelin, where it maybe suggested it was the most specific with the y-intercept, we see here it's also very sensitive to the iron storage protein. So maybe that specificity isn't quite there. Interestingly, the R2 star correlation was the lowest, where R2 star is typically used to look at iron content. It needs to be taken with a grain of salt because this is just an iron storage protein. It isn't necessarily indicative of the iron loading of the cell. So some limitations of this study: it was performed on a single healthy marmoset, and these relationships shown could be disrupted in pathological conditions. Additionally, we assumed a linear relationship between the MRI and histology, mainly due to the limited ROIs used. We're hoping that once we have the full data set together and we can either do voxel-wise analysis or include more ROIs, we can investigate some maybe non-linear relationships that could exist. So in conclusion, we investigated MRI contrasts, and they're all highly correlated with myelin, and that MRI metrics presented differing degrees of sensitivity to the other histological metrics. And this might be important for investigating certain neuropathologies, something like in Parkinson's disease, where you end up with disruptions to storage proteins. You might start to see a divergence of the mutual correlation spatially. So future work's going to look to combine what I presented today with some other stains that we've also acquired. So myelin basic protein, we have microtubule stain, as well as neurofilaments, as well as some nice diffusion data. So here are 2 calculated maps from some B-tensor encoded diffusion data, also in the same marmoset. So we're looking to bring all of this together. And once all of the MRI and the histology is processed, we're hoping to package it together and make it openly available to other researchers to use. And that is it. So I'd like to thank all my collaborators. There's a ton of work and a ton of expertise needed to get this together. So I appreciate all of their help in this. Thank you.